Hello everyone. So let's see the contents of our topic proteins. It will include introduction, types and classification, synthesis, metabolism, chemical nature, biological role, sources, disease and disorder, and facts. With introduction, proteins are large biomolecules and macromolecules that consist of one or more large chains of amino acid residues. They are a linear chain of amino acids. The individual amino acid residues are bonded by peptide bonds. Proteins only exist for a certain period and are degraded and recycled by the cells machinery through the protein turnover, and they can exist for minutes or years with an average lifespan of 1 to 2 days in mammalian cells. So let's see the types of the proteins. So proteins are classified according to the structure. The primary structure, which is a polyimide. The secondary structure, it regularly repeating local structures which consist of the alpha helix, the beta sheets and turns. And the tertiary structure, that is the overall shape of a single protein molecule, the spatial relationship of the secondary structures to one another. And the tertiary structure is generally stabilized by non-local interaction. The term tertiary structure is often used as synonymous with the term fold, and the tertiary structure is what controls the basic functions of the protein. The quaternary structure, that is the structure formed by several protein molecules, that is the polypeptide chains, and the quaternary structure is dependent on transient yet essential macromolecular interactions that occur inside the living cells. Now classification of proteins. So it is based on structure. It contains fibrous proteins, globular proteins and intermediate proteins. Classification based on composition, simple proteins and conjugated proteins. Classification based on functions, structural proteins, that is enzymes and hormones, pigments, transport proteins, contractile proteins, storage proteins and toxins. Now let's see the protein synthesis. So there are five major stages of protein synthesis. Stage 1 is activation of amino acids. This reaction is brought about by the binding of an amino acid with ATP. The step requires enzymes called amino-acyl RNA synthetases. Due to this reaction, amino acid and adenosine triphosphate mediated by above enzyme. And the stage 2 is transfer of amino acid to tRNA. The amino acid AMP enzyme complex formed reacts with specific tRNA. Thus, amino acid is transferred to tRNA. As a result, the enzyme and AMP are liberated. The stage 3 is initiation of polypeptide chain. Charged tRNA shifts to ribosomes. The ribosome consists of structural RNAs and 80 different proteins. The information for the sequence of amino acids is present in the sequence of nitrogenous bases of mRNA, and each amino acid is coded for 3 letters word of nucleic acid. The initiation of polypeptide chain in prokaryotes is always brought about by amino acid methionine, which is regularly coded by the codon AUG, but rarely also by GUG for valine as also initiating codon. The stage 4th is chain termination. The termination of polypeptide is signaled by one of the three terminal triplets, that is the codons in the mRNA. They are also called stop signals, that is UAG, UAA and UGA. At the time of termination, the terminal codon immediately follows the last amino acid codon and the subunits of ribosomes gets dissociated. The fifth step is the protein translocation. The two classes of polyribosomes have been identified. That is, for free ribosomes, termination of protein synthesis leads to release of completed protein into cytoplasm, and on the other hand, in membrane-bound polyribosomes, polypeptide chain grows on mRNA and is inserted into the lumen of endoplasmic reticulum membrane. Now let's see the protein folding. Once synthesis of the polypeptide chain is completed, the polypeptide chain folds to adapt a specific structure which enables the protein to carry out its functions, and the basic form of protein structure is known as the primary structure, which undergoes certain changes to form the secondary structure, the tertiary structure, and the quaternary structure, as we can see in the figure. And finally, some proteins may adopt a complex quaternary structure, that is an example of hemoglobin. Now we will see the metabolism of amino acids. The amino acids undergo certain common reactions like transamination followed by deamination for the liberation of ammonia. The amino group of the amino acids is utilized for the formation of urea, which is an excretory end product. The carbon skeleton of the amino acids is first converted to keto acids, which meet one or more of the following fates. Now, as you can see in the figure, the main sources of the amino acids are dietary protein and the body protein, which undergoes transamination to form the keto acids. The transfer of an amino group, that is NH2, from an amino acid to a keto acid is known as transamination, and this process involves the interconversion of a pair of amino acids and a pair of keto acids catalyzed by a group of enzymes called transaminase. Silent features of transamination. All transaminases require pyridoxal phosphate, a coenzyme derived from vitamin B6. Specific transaminases exist for each pair of amino acid and keto acids. There is no free NH3 liberated, only there is transfer of amino group, and the transamination is a reversible process. Transamination is very important for redistribution of amino groups and production of non-essential amino acids as per the requirement, and it diverts the excess amino acids towards the energy generation. The amino acids undergo transamination to finally concentrate nitrogen in glutamate. All amino acids except lysine, threonine, proline, and hydroxyproline participate in transamination. Mechanism of transamination. Transamination occurs in two stages: transfer of the amino group to the coenzyme pyridoxal phosphate to form pyridoxamine phosphate. The amino group of pyridoxamine phosphate is then transferred to a keto acid to produce a new amino acid, and the enzyme with pyridoxal phosphate is regenerated. The above figure represents the involvement of pyridoxal phosphate in the transfer of amino group. The above figure represents the formation of enzyme PLP skif base and amino acid PLP skif base, and when the amino acid binds, the enzyme separates. All the transaminases require pyridoxal phosphate, a derivative of vitamin B6, and when an amino acid comes in contact with the enzyme, it displaces lysine and a new skif base linkage is formed. The amino acid PLP skif base tightly binds with the enzyme by non-covalent forces. Deamination. The removal of amino group from the amino acids as NH3 is deamination, and it results in liberation of ammonia for the urea synthesis. Deamination may be either oxidative or non-oxidative. The oxidative deamination is the liberation of free ammonia from the amino group of amino acids coupled with oxidation, and it takes place mostly in livers and kidney. The purpose is to provide NH3 for urea synthesis and D-keto acids for a variety of reactions, including the energy generation. This figure represents the oxidative deamination of amino acids, where L-amino acids is transformed to alpha-keto acids with the help of L-amino acid oxidase. Non-oxidative deamination. Serine, threonine, and homoserine are the hydroxy amino acids, and they undergo non-oxidative deamination to form respective alpha-keto acids, which are catalyzed by dehydratases. The sulfur amino acids, namely cysteine and homocysteine, undergoes deamination coupled with desulfhydration to give keto acids, and the enzyme histidase acts on histidine to liberate NH3 by a non-oxidative deamination process. Urea is the end product of protein metabolism and it accounts for 80 to 90% of the nitrogen containing substances excreted in urine, and it is synthesized in liver and transported to kidneys for the excretion in urine. The urea cycle that was elucidated by Hans Krebs and Kurt Henseleit in 1932, and it is a five-step cyclic process with five distinct enzymes. The first two enzymes are present in mitochondria while the rest are localized in cytosol. The above figure represents the urea cycle. Step 1. Synthesis of carbamoyl phosphate. Carbamoyl phosphate synthase 1 of mitochondria catalyzes the condensation of NH4 plus ions with CO2 to form carbamoyl phosphate, and this step consumes 2 ATP and it's an irreversible process. Step 2 is formation of citrulline. Citrulline is synthesized from carbamoyl phosphate and ornithine by ornithine transcarbamylase, and ornithine is regenerated and used in urea cycle. Step 3 is formation of arginosuccinate. Arginosuccinate synthase condenses citrulline with aspartate to produce arginosuccinate, and the second amino group of urea is incorporated in this reaction. This step requires ATP, which is cleaved to AMP and pyrophosphate. Step 4 is cleavage of arginosuccinate. Arginosuccinase cleaves arginosuccinate to give arginine and fumarate, and the fumarate liberated here provides a connecting link with TCA cycle and gluconeogenesis, etc. Step 5 is formation of urea. Arginase is the fifth and final enzyme that cleaves arginine to yield urea and ornithine, and the ornithine regenerated enters the mitochondria for its reuse in the urea cycle, and the arginase is mostly found in the liver while the rest of the enzymes in the urea cycle are also present in other tissues. Overall reaction and energetics. The urea cycle is irreversible and it consumes 4 ATP, where NH4 plus CO2 plus aspartate plus 3 ATP are used and they are converted to form urea, fumarate, 2 ADP, 2 PI, AMP and PPI. Regulation. Carbamoyl phosphate synthase 1 catalyzes the rate limiting step of the cycle and is stimulated by N-acetyl glutamate, and the rate of urea synthesis in liver is correlated with the concentration of N-acetyl glutamate. The remaining four enzymes of the urea cycle are mostly controlled by the concentration of their respective substrates. Metabolic disorders. The metabolic defects associated with each of the five enzymes of the urea cycle have been reported. All the disorders invariably lead to build-up in blood ammonia, that is hyperammonemia, leading to toxicity, and other metabolites of urea cycle also accumulate. The clinical symptoms associated with defect in urea cycle enzymes include vomiting, lethargy, irritability, ataxia, and mental retardation. Now let's see the metabolic defects in the urea cycle. The hyperammonemia type 1, the enzyme involved is carbamoyl phosphate synthase 1. The hyperammonemia type 2, the enzyme involved is ornithine transcarbamylase. The citrullinemia, where the enzyme involved is arginosuccinate synthase. The defect arginosuccinic aciduria, the enzyme involved is arginosuccinase, and the defect hyperarginemia, the enzyme involved is arginase. Chemical nature of proteins. Hydrolysis, that is breaking of polypeptide bonds by enzymes and by acid. Denaturation, that is disruption of non-covalent bonds by heat, mechanical agitation, detergents, pH change that leads to disrupt salt bridges, and solvents that leads to disrupt hydrophobic interaction. The unfolding of polypeptide chains can be caused by agents such as heat and urea that do not cause hydrolysis of peptide bond. Secondary and tertiary structures are destroyed by denaturing agents but is left intact. The process of renaturing a denatured protein after removing the denaturing agent is renaturation. An amino acid's average molecular weight is 110. The molecular weight of protein is determined by multiplying its amino acid content by 110, and the molecular weights of the proteins are different because of their amino acid compositions, and most proteins have a molecular weight of 5000 Daltons or more. Now we will see the biological role of proteins. The binding proteins typically interact non-covalently with their specific ligands. The transport proteins are one class of the binding proteins, and they include membrane proteins that transport substance across the membranes, as well as soluble proteins that deliver specific nutrients. The scaffold proteins are a class of the binding proteins, and the catalyst proteins mediate almost every metabolic reactions. The biological importance of proteins has a nutritional role which provides the body with essential amino acids, the catalytic role, the hormonal role, and the defensive role, where the antibodies that play an important role in the body's defensive mechanisms are the proteins in the nature. These are the various biological importance of the proteins. Many proteins are protective as well as supportive. The hemoglobin is a protein which carries O2 in the blood. The actin and the myosin are the contractile proteins. The amino acids are converted to other nitrogenous substances of great physiological importance. Let's see the various sources of the proteins. Powder protein can come from a variety of the sources like eggs, milk, and plants, that is soybeans, peas, and hemp. Some protein powders contain protein from multiple sources. For instance, a vegan option might include protein derived from peas, pumpkin seeds, sunflower seeds, and alfalfa. 10 terrific sources of lean proteins include fish, seafood, skinless white meat, poultry, lean beef, skim or low-fat milk, the low-fat yogurt, fat-free or low-fat cheese, eggs, lean pork, and beans. This figure shows us various types of plant sources of the proteins, which includes tofu, lentils, and the animal sources of the proteins, which include sardines, eggs, milk, etc. Now we will see the protein diseases. The protein misfolding diseases includes the conditions where a protein fails to fold correctly, that is cystic fibrosis, and is not stable enough to perform its normal function, that is many forms of the cancer, fails to be correctly trafficked, that is familial hypercholesterolemia, and the next is forms insoluble aggregates that deposit toxically, that is neurodegenerative diseases, Alzheimer's, type 2 diabetes, Parkinson's, and many more. This figure shows various protein misfolding diseases that includes disordered protein aggregate, amyloid, enhanced degradation, and trafficking defect. Some diseases and proteins responsible for them. Alzheimer's disease, where the protein that is responsible is amyloid B peptide. The type 2 diabetes, the protein responsible is isolate amyloid polypeptide. The sickle cell disease, where the protein responsible is hemoglobin. Disease related to protein excess, which includes heart disease, cancer, adult bone loss, that is osteoporosis, and kidney disease. Protein deficiency leads to loss of muscle tone, confusion, irritability, etc. And protein excess leads to acidosis and dehydration, constipation, musculoskeletal issues, etc. Facts about proteins. A protein is a naturally occurring and extremely complex substance. It is a Greek word and it comes from the word proteos, which means primary or the first rank. The proteins are present in all living organisms and include many essential biological compounds. Proteins are very essential to all life forms and you couldn't grow or heal without it. They have very short lifespan as there are about 1 lakh different types of the proteins in human body, but the lifespan of these proteins is totally to 2 days. Hair is made up of a protein called keratin, which forms a helical shape. Gluten is a protein. All enzymes are proteins, but all proteins are not enzymes. In our body, the enzymes like trypsin, pepsin helps in the protein digestion, and each gram of the protein gives us 4 calories. Thank you so much for watching.